Hello, everyone. My name is Dan Tempest, and I'm here to do my educational presentation on the cell membrane structure and the function of the transport across the cell membrane. So here we go. All right, so these are learning objectives that I use in my AP Biology class. So you can see they're broken down into five categories: the plasma membrane, the membrane permeability, membrane transport has two, and then we have facilitated diffusion and the mechanisms of transport. Um, those are what I go through in this lesson particularly, or these set of lessons particularly. So I always like to start out with this picture, and I introduce them to this term called the fluid mosaic. So what I always like to do first is I just ask them the question, what is a mosaic? And usually somebody can come up with that it's a lot of parts making a whole, and then they can make that connection to this picture. Um, so then why do we call it a fluid mosaic? That is when I'll bring up terms like cholesterol, and I'll point out a cholesterol molecule on the diagram. I can also talk about how the lipid tails of a phospholipid are sometimes unsaturated, and that can help with the fluidity of the membrane. And we just talk about that for a little bit, and I talk about how cholesterol really aids at specifically extreme temperatures in increasing the fluidity, or sometimes at really hot temperatures, even stabilizing the membrane can help. So I like to use this picture, and I think students can make that connection to that fluid mosaic term. So then I get into the structure of a phospholipid. They are the most abundant lipid in the plasma membrane by far, and they have this physical characteristic of being amphipathic. So being amphipathic means you have two opposite parts. So essentially we have a phosphate head, and I point out that that is attracted to water, so we bring up this term called hydrophilic, meaning water loving, and we break apart that term. We break apart hydro, and they usually always say water as it relates to hydro, and then I bring up this term philic, and we tell them that that means loving. So water loving. Then we have hydrophobic regions, which is the lipid tails, and we talk about this term hydrophobic, and hydrophobic is a term that they usually do a really nice job with. Hydro, again, meaning water. Phobia, they can usually come up with as a fear. So these lipid tails are not going to want to mix with water. So we talk about those opposite parts, that phosphate that needs to be able to interact with that water, and then the lipid tails repelling that water. And we talk about how that makes or allows a cell to create a water barrier. Okay, so then I really like this picture here. This picture shows that water is both inside a cell and outside a cell, so that's why we need that phospholipid bilayer. So again, the phosphates on each side point toward the water, and I really like this picture up here that shows the water molecules, and you have the phosphate head interacting with that water molecule. And then it shows no water molecules in by the lipid tails, and so we talk about how, again, reminding them that polar and nonpolar things don't like to mix, just like we did already with the water properties, and we talk about it then too. So really nice picture, again, showing those water molecules interacting with the phosphates and not the lipid tails. All right, so then I like to bring in protein structure. So we've already, in AP Biology, when I go through the cell membrane and transport, we've already done protein structure. So I really like to bring in this concept here, kind of reiterating that point and reviewing. Anytime I can make connections, we really try to do so. So when we talk about the primary structure of a protein, we're talking about the amino acids and the order of those specific amino acids, and that really controls the rest. So then we look at secondary structure of an of proteins, and that's going to be those repeated configurations, such as in this picture we have the alpha helices. So those spiral coil looking shapes, and that's going to be really non-polar in nature. So then I point out that it's no coincidence that that is in that non-polar core next to those lipid tails, creating, again, that water barrier. And then we point out that out here we would probably have some polar amino acids, or at least polar R groups would be pointing up there. And again, because that part of this particular protein needs to be able to interact with that water. So I wouldn't just come out and tell them that right away. What they would be introduced to this question. So this question would just be up on the board, and I would hopefully they could come to at least parts of that. And anytime I can fill in the gaps, then I would. So then we go into the 6 major functions of membrane proteins. And so we look at here, we have transport. So we're going to have transport of solutes across the membrane. We're going to have enzymatic activity. So we're going to make some reactants into some products, catalyzing probably, and we're going to have some signal transduction then. We can see a signal molecule binding to a receptor protein and starting that transduction. We're going to have cell-to-cell recognition, and we'll talk more about that, but later in this, when they do immune system, this is really important in the immune system, recognizing self versus not self, for instance. Intracellular joining with gap and tight junctions, we'll talk about that. And then interactions with the cytoskeleton and how that aids in the structure of a cell. So we'll talk about that briefly. This is when I really like to break this down. So as I mentioned, one of those functions, specifically, if I go back with the cell-to-cell recognition, is really important when we go with the immune system. If I go back again, this signal transduction, really important as it relates to the nervous system with neurotransmitters and binding to those ligand-gated ion channels. So I really like to bring in, as well as the endocrine system with hormones binding to their receptor molecules on cell membranes quite often. So I really like to bring in connections at this point, because most of the time students, when they come to my class, have heard of these systems, or at least are able to relate somewhat to these. So I like to have them do this activity where they're going to connect the structures of the membrane to these body systems. And I give them some pages out of their textbook to look through. So what I would do at this time is I would break them into groups, and I probably have 2 groups in each class doing each one of these. So I have 2 groups doing nervous system, 2 on immune system, and 2 on the endocrine system, and trying to help them just to make connections. So they don't have to go through that whole chapter. What they're trying to do here is they're making connections to that cell membrane, and how that cell membrane is really important to the functioning of these particular body systems. So now I'm going to look at the really, why is it important? We talked a little bit about the structure now and how that really can help a cell create a barrier. But if we look at really, why is that important? I really love this picture that is from biologyaspoetry.com. We have to be able to allow certain things in and certain things out in a cell. So if we look at this, we have certain molecules are able to freely pass through, and I'll get into those characteristics, but this is an example of simple diffusion here. So we have oxygen, carbon dioxide, other things like steroids can do this, but you can see this huge arrow, and that just means that they can freely pass through this membrane. Then we have other things, usually because they're small, but they are hydrophilic, so they're still water-loving, so they're not going to want to pass through that membrane really freely due to those chemical structures of being polar, but they can, because they're so small, still at small amounts pass through that membrane. So we'll talk a little bit about that, because sometimes they have a misconception that water can't pass unless it has a protein. So I do like to clear that up. Then we have these very charged molecules like sodium and other things, or large molecules like glucose, not able to pass at all. Again, protein, starches, again, being very large, not being able to pass. So it's really important for them to make the connection that we have to be able to allow certain things in, but also allow certain things out. And related to that, we have bulk transport. One of the things that cells need to be able to do is secrete protein products a lot of the times out. So we've already talked about the endomembrane at this time, the endomembrane system, and how we can secrete proteins through that. So we look at this picture again, reminding them of that, making proteins and lipids in the endoplasmic reticulum through transport vesicles, allowing that to go to the Golgi. But if you look at the sidedness of each one of these molecules, it allows for those vesicles to be able to fuse. and specifically we look in here, fuse outside, so that we just talk a little bit about that sidedness of the membrane at this time. So then we go into hydrophobic and hydrophilic molecules. So hydrophobic molecules such as steroids and testosterone, we talk about how they can freely pass through the membrane and why, because they are able to interact with that hydrophobic core with the lipid tails, versus polar molecules do not freely cross. So they're not rapidly. That doesn't mean they can't cross. Again, we mentioned water being small can pass through at small intervals. Glucose, on the other hand, being polar, cannot pass through, and it's also large, cannot pass through at all. So we talk about kind of different molecules and how some of them can cross, some of them can't. Transport molecules. So what about those molecules that can't cross? How do we get them in? How do we get them out? And we talk about these transport proteins. And I really like basketball, so I always bring up the facilitator. Point guards are the facilitators. So I talk about how point guards are like the facilitator proteins of the cell. They have to try to get other people involved. And so they're allowing these molecules to get into or out of the cell. And we specifically talk about aquaporin, which is the protein that allows water to pass. Now getting into again, some of these are examples of passive transport. So we talk about, in this underlined and bolded and italicized, no energy investment is needed in passive transport. So then we go into two examples of passive transport. So we talk about simple diffusion. Again, those molecules that can freely pass, they don't need a transport protein at all, facilitator protein, versus facilitated diffusion, which is when molecules do or move a lot better, at least, if they have that transport protein. So osmosis is a good example of that, using water using that aquaporin. So simple diffusion, I'd like to go through that next. So simple diffusion, water moving from high to, or not water, but movement of concentration from high to low. And we get this diagram here. We have molecules of dye moving through a membrane, and they're able to pass through, and eventually they reach this equilibrium, but we show them high concentration to low. I really like the model where I have the Febreze up in front of class, and I'll spray it, and it's really pungent to both me and the people around me. But once we reach the corners of the room, it's not going to be nearly as pungent. Movement from high to low. Facilitated diffusion, again, requiring that facilitator protein. So we talk about channels and carrier proteins, slight differences where the carrier has to change slightly in shape. And these are really aiding in the speed of that movement for molecules that might struggle to pass through that hydrophobic core or are really big. And then that last part is the opposite of passive transport. We need to hit that active transport, where now if you're being active, you have to have energy. And so active transport uses energy. Specific example, the sodium-potassium pump, which is going to allow us to return to that resting membrane potential after an action potential, is a good example of this active transport. But the key thing here is two things. One, it uses energy. Two, goes from low to high. So we're moving against that concentration gradient, which is why we need that energy. So we hit that. Then I like to give them this as a warm-up. Usually this is probably the next day. And I don't have these obviously labeled when I give them as a warm-up, but I'm going to ask them real simple, or maybe as an exit slip at the end of the class. Label these movements. So if it's going directly through the membrane, we got simple diffusion. If we have movement through a transport protein, a facilitator protein, it's going to be facilitated diffusion. Both of those go from high concentration to low concentration. Therefore, we consider this passive transport. No energy needed for that. And if we look at this, we're looking at these diamond shapes. They're moving, in this case, from low to high. And here we have ATP being involved, supplying that energy needed for active transport. All right, so at this point, I would probably release them to review So I give them 3 options. I really love Mr. Anderson with Bozeman biology videos for AP. He's phenomenal. I think a lot of kids really enjoy and they love how in depth he goes, but also making it simple, maybe in ways that sometimes I struggle with. So one on cell transport, one on the cell membrane structure in general, and then also this one was through GCU, their library, which I found a little bit more advanced on the cell membrane. So if we want some high-end kids that have some extra time, I'll have them look at that. One activity that I really love to do in this chapter, great review, is a poster modeling. So I used to do this in posters. Now I have the markers that can write on the lab tables, which kids, for whatever reason, they love. So I have them right on their tables, do some poster modeling. They first have to plan the entire thing out. So I would give them a sheet with all kinds of different things that they're going to have to include, different movements, different structures within the cell membrane. They have to model all of that on a poster project. So I have them plan it out, then they're going to have to draw it out. So they have to identify the phospholipid bilayer, labeling phosphate heads and lipid tails. They're going to have some facilitator proteins, some glycolipids, glycoproteins, cholesterol, and then a peripheral protein. Those are just some structural components. They have to model the movement of sugar, so showing some facilitated diffusion. They have to show the movement of some lipids, so we have some simple diffusion. Water movement, so we have some osmosis on there. And then we have an example of active transport, specifically with sodium. And then I have them label some passive versus active transport on that poster model. So then another thing that I really like to do is an AP class. I have always a lot of high-end kids. A lot of them go in the medical field. Some of them are going engineering, but they're all mostly science kids. So I like to have real-world applications. So how is the world using this? And I think they really love this activity every year. We do. How are the properties and characteristics of the cell membrane used in engineering? So I found in a specific book that I found, they have all these engineering and how we use biomolecules such as a phospholipid, for example, in engineering practices. So it's a small part of this, but they would look through this link and they would have to tell me, how is that cell membrane components being used today in engineering? And then here we have my references, and I hope that was helpful today. Have a great day.